Okay, now we're going to begin looking at the structure of a nephron. And you're either going to be watching this video before we had a chance to discuss it in class and draw it on the whiteboard, or after. And if it's before you had a chance to draw it out, this is going to be a little tricky. If you've already experienced my drawing in class, it's going to be largely a review. But we have to kind of look at two things separately. There's the blood flow, and then there's the actual nephron and its tubules. So let's first start at blood flow. If you recall from previous lectures, the renal artery has to divide and branch into smaller arteries and arterioles. And before you get to a nephron, one of them is known as the arcuate artery. And that's this guy right here. And off of that arcuate artery, there's an artery, or really an arteriole, that extends out into the cortex of the kidney. You'll notice on this drawing, this outer region that's a little bit lighter is the renal cortex, whereas this bottom area, this deep area, is the renal medulla, the part that's found in the renal pyramid. So as this arteriole extends out into the cortex, it gives off little arterioles to each of the nephrons. And those little arterioles are called the afferent arterioles. So that's a really important blood vessel right there, the afferent arteriole. And blood's going to go into the glomerulus, which is this kind of looped up set of capillaries. The glomerulus is a fenestrated capillary, so it allows a little more filtration than a normal capillary. And it's all looped up. Surrounding that looped up capillary is the collecting portion of the nephron, called Bowman's capsule. So Bowman's capsule surrounds the glomerulus, and together those two structures are called the renal corpuscle. So fluid and solutes are being filtered out of the blood, collected into this capsule, so they can then travel into this tubule portion of the nephron. Anything that does not get filtered out leaves the glomerulus in what's known as the efferent arteriole. And that efferent arteriole, you always notice it's much smaller than the afferent. It's smaller because about 20% of the volume of blood gets filtered out. So therefore, there's 20% less volume coming out this efferent than what came in using the afferent. That efferent arteriole then starts to branch, and it forms all these capillaries. All these capillaries that surround the tubule are known as peritubular capillaries. And those are going to become important for us when we get into how this nephron functions. So that's the blood flow. But now we've got to follow what got filtered out of the blood, and that's called filtrate. Filtrate is what got filtered out of the blood, collected into Bowman's capsule, and now it's going to travel through the tubule portion of a nephron. You'll notice down here is kind of a flow list. So you can refer to this list as we follow it through the nephron. So once it gets collected, the filtrate enters what's called the proximal convoluted tubule. Proximal because it's closest to Bowman's capsule. Convoluted means it's twisted. So it kind of loops around and twists back and forth. Then it turns into the loop of Henle, or the nephron loop. And the loop has a descending limb because the filtrate descends down into the renal medulla. Then there's an ascending limb where it comes back up. Then it extends into another convoluted region called the distal convoluted tubule, then into what's known as a collecting duct. Collecting ducts, you'll notice, get filtrate from many nephrons. So you can see that there would be one here. So a nephron would be dumping its filtrate in there, another one here, another one here. So a collecting duct collects filtrate from many distal convoluted tubules. Then, if you follow the collecting duct down, you'll notice that many collecting ducts will merge together into what's called a papillary duct. And then that papillary duct, which is way down in the point of the pyramid, is where the urine kind of drips out. And then there's going to be that minor calyx collecting those little drips of urine. So those are the most important parts of a nephron. When we get to nephron physiology, we have to know what happens along the way. We already know one step is to filter. This is where filtration happens. Filtration, if you recall, we've used that word in the past. But filtration is when fluid and solutes move out of the bloodstream, and in this case, it gets collected into the Bowman's capsule. The next thing we need to learn is what happens to this filtrate as it flows through these different parts of a nephron. This is an expanded view of the renal corpuscle. So recall, a renal corpuscle is the glomerulus surrounded by Bowman's capsule. So this is just an enlarged version of that part of a nephron, where we see that there's an afferent arteriole bringing blood in to the glomerulus, which has these loops, these capillary loops, and then the efferent coming out. So these arrows represent fluid that was filtered out of the blood. That fluid is largely water and small molecules. So the glomerulus acts as a filter, and it's largely based on size. So small molecules get filtered out. So if you think of small molecules, that would include water, that would include ions, sodium, chloride, potassium, calcium. It would include small molecules like glucose, amino acids, some metabolic wastes. That all gets filtered out. Now the problem with that, if you think about it, some of that is waste and you want to get rid of it. Others you don't want to get rid of. You don't want to get rid of all your electrolytes. You don't want to get rid of glucose and amino acids. So the next thing to do along the tubule is to start to reclaim a lot of those small molecules that you don't want to eliminate. So after filtration occurs, this nephron, starting at the proximal convoluted tubule, starts to reabsorb things, starts to reclaim small solutes from the tubule back into the bloodstream. And that's called reabsorption. You'll notice that on the glomerulus there are some cells called podocytes. Podo means foot, cyte means cells, because they have little feet, they call these things. They're pedicels. That forms part of the filtration apparatus, if you will. There are also some other cells in there, the mesangial cells. They're going to help regulate filtration. Filtration has to be tightly regulated. The driving force of this process is blood pressure. So the reason why fluid gets pushed out is blood pressure. And if blood pressure goes really, really high, you don't want to filter too much. So there's got to be mechanisms to decrease the amount of filtrate. You also want to be able to increase filtration. So there's a lot of adjustments and control of what we call GFR, glomerular filtration rate, which is basically how much filtrate do you produce in a certain unit of time. You'll notice that there's actually part of that tubule that extends back here. This is part of the ascending limb or distal convoluted tubule. But if we go backwards and look at the previous picture, notice that right here, there's part of either that ascending limb or part of the distal convoluted tubule, which is back very close to this part of the capsule or corpuscle. So that's why this little guy is shown here. So this is the renal corpuscle, and this is the site of filtration, removing fluid and solids from the blood, collecting it into the Bowman's capsule, and then it enters the tubule. This drawing is just to introduce us to the steps that occur during renal physiology. This is not drawn anatomically, so this is the same thing. It's a nephron, but it's not drawn the correct way. It's just a simplified version so we can understand the three processes that occur. First process we already mentioned. It's called glomerular filtration. The green arrow represents filtration. Fluid and solids from the blood plasma, which means we don't take any cells. Red blood cells don't get filtered. White blood cells don't get filtered. Platelets don't get filtered. It's just fluid and small solids. They move out of the glomerulus. They get collected into the capsule, glomerular capsule or Bowman's capsule. Then they enter the tubule. And that filtrate is going to start to flow through the renal tubule or the nephron's tubule from the proximal to the loop to the distal, then the collecting duct. Now, along the way here, we're going to start to reclaim what needs to stay in the body. That process is called tubular reabsorption. That's shown as this arrow. It's taking fluids, so we're going to start to reclaim water because we don't want to lose too much water, and other solids, and return them to the bloodstream. This is all done using transporters. There's a number of different transporters which transport useful solids like water, like sodium, like chloride, like potassium, and put them back into the blood. Then there's another way to get rid of stuff. It's called secretion. It's similar to filtration in that you're moving things out of the blood into the nephron. The difference is it happens along the tubule. It doesn't happen at the corpuscle. But there's transporters that can help us get rid of things that we didn't get out of the blood. So notice that this blood, after it leaves the glomerulus, and then it enters the efferent, it turns into the peritubular capillaries. This represents a peritubular capillary. Well, what if we didn't get rid of a waste when we filtered? There's another chance to get rid of it, and that's through the process of secretion. So secretion moves molecules from the blood into the tubule. And it's another way we can get rid of waste or excessive amounts of solids. So then whatever's left in the tubule is going to be urine. So I always think of this process as simple math. You filter, so whatever you filter enters here. Then you subtract what you reabsorb, and then you add in what you secrete, and what you get is urine. So you make filtrate during filtration, you take back what you reabsorb, and you put in what you secrete. And that is what urine is. In the next video, we're going to talk a little more about filtration, what gets filtered, how we regulate that filtration. Then we'll move into the tubule and start looking more at the next two steps, the reabsorption and secretion that occurs.